Dr. Neil Oxtoby is a Principal Research Fellow and a UK Future Leaders Fellow in the University College London Centre for Medical Image Computing and in the Department of Computer Science. He notes that his passion is improving healthcare through computer science that includes close relationships with industry in order to maximize impact and uptake, particularly in dementias. So more broadly, he jointly leads the UCL Progression of Neurological Disease called POND group and has a sustained track record of developing novel computational approaches that reveal new understanding of disease progression. His group leads and has led multiple international consortia and medical computing initiatives that are motivated to provide data-driven solutions to healthcare challenges and chronic diseases. Okay, Dr. Oxtoby runs the biennial POND conference and is the data science lead of the International Network for Deep Phenotyping in Dementia. Finally, he is a founding member of the new iStart PIA on artificial intelligence for precision dementia medicine. He notes that he is a former physicist and currently enjoying the challenges of modeling the human brain. I would be remiss if I did not include that Dr. Oxtoby describes himself as a scientist by day and a musician by night. I think that's a very interesting point to make for our speaker today. Christina has actually included in the chat a link to the music. So we will listen to the science first before we go to the music. And we are very grateful for your being here for your talk today. And the title is Data-Driven Disease Progression Modeling for Research and Possibly for Healthcare. So thank you, and I'll let you take it away.
Thank you very much. It's great to be here. Yes, so my name's Neil, and I got in trouble from my physics colleagues for describing myself as a former physicist. I said, once a physicist, always a physicist. So I perhaps should update that part of my my bio sketch. So I lead the POND group along with Professor Danny Alexander, a computer scientist, in the Center for Medical Image Computing, which bridges computer science and medical physics. We do lots of stuff from new models for image segmentation and registration all the way through to image-based modeling, like disease progression modeling. And you saw this slide flashed up earlier, those who were here before the recording started. This is the entertaining title for my fellowship. It's a 4+3 year fellowship to try to put the research funders' money where our researchers' mouths are. You read a paper and it says this is going to be useful for Alzheimer's disease clinical trials, and then no one goes on and does it. They just publish their next Nature Medicine paper and walk away. Not that I've got one of those. So the quest is for supermodels, which are disease progression models, and I'll explain that. And the drugs reference is about how it helps with clinical trials. And it came from me being sick and tired of reading these papers that never delivered on their promises. So an overview. Essentially, you can distill the talk down to three things. I'm going to talk about Alzheimer's disease with a focus on how heterogeneous it is. Tell you about disease progression modeling, and the key takeaway there is the pseudo time, where it estimates the time, the disease time, at the same time as estimating the trajectories of disease progression. And then I'll talk specifically about applications of disease progression modeling to clinical trials and concepts like enriching clinical trials with people who might make the clinical trial a success, depending on the outcome of that trial, the desire of that, the goal of that trial. Probably know this, let's do it anyway. Alzheimer's disease is actually defined post-mortem by histopathology, the pathology that's seen in the brain at post-mortem. However, it's become a syndrome, a clinical syndrome, memory-led clinical syndrome, but I've emphasized here the etc. There's more time memory loss in the syndromic phase. And the syndromic phase doesn't happen until right at the end. There's a long pre-symptomatic period. Seems to be that there are decades of pathology that build up before you see symptoms and before you might seek healthcare. The challenge here then is to track that long pre-symptomatic period, understand it, and then find optimal windows for intervention. But that's complicated by the heterogeneity that we see. And there's heterogeneity in the syndrome, as I mentioned, more than just memory. There's heterogeneity in the onset, age of onset, the speed of progression, the severity of the disease in both the syndromic and pre-syndromic phase. And in the pathology itself, you probably all know, but I've got colleagues who do histopathology at Queen Square here in London, and they've seen heterogeneity between family members with familial mutations for Alzheimer's disease. So, notwithstanding the exciting news of last week at CTAD, treatments for Alzheimer's, what exists? Well, we've already talked about the amyloid cascade hypothesis for a long time. The pluses for that is that there's plenty of supporting evidence. It does seem to be the way that things are going. It does seem to be an amyloid-led disease. What has been posed as evidence against the amyloid cascade hypothesis and our understanding of the cause of Alzheimer's is that anti-amyloid therapies have not been proving efficacious in large clinical trials. And this statement stands, despite the impressive results from lecanemab, the Clarity AD trials last week. That's a very small number of successful trials over a decade's worth of trialing, hundreds of therapies across multiple phases of disease of clinical trials. So why have they been failing? Potentially, you could imagine that you're recruiting people at the wrong time. Remember, this long pre-symptomatic phase of pathology could be too late. So we could need to be thinking about prevention versus cure, and there is some effort in prevention, clinical trials for prevention. We could be picking the wrong people. So there could be patients at risk of Alzheimer's disease or with mild syndromic Alzheimer's who are not going to respond in that clinical trial period, and that could wash out the treatment effect. Likewise, the treatment might take longer to work, so the trial might be too short, insufficient duration. The wrong endpoints, we could be picking up the wrong biology or not picking up clinical benefit for various reasons, or controversially perhaps, the amyloid hypothesis could be wrong. We could have the wrong strategy. And there's some reading material on the rest of this slide about these talking points. So how do clinical trials try to find the right people at the right time for a trial? Well, there's a few phases of the clinical trial process, recruitment process. The screening, and then you look to exclude individuals for various reasons. You actually choose to screen people in based on some criteria, and then you assess their risk of decline right about the same time. So typically screening is done with clinical measures, something fairly simple or some kind of a very simple test of risk factors like ApoE4, or more recently, there's some plasma biomarkers that are looking quite promising. Otherwise, you can do various demographics that increase your risk and cognitive tests. That will get you in the door for a clinical trial. And you might have some incidental findings on MRI that would exclude you. You could have some other problem with your brain that's causing the problems in cognition. You could have vascular pathology that might be a good reason not to take an experimental treatment. Typically, that's assessed with structural imaging like MRI. And then in the more recent trials, especially to make sure these particularly the anti-amyloid and anti-tau treatments, you would take a PET scan or a CSF measure of these proteins and screen someone in based on some threshold of tolerance for that. And the risk of decline can be assessed across multiple modalities in terms of neuronal loss or synaptic dysfunction, depending on the trial you're looking at. There's a couple of papers down here in Alzheimer's Research and Therapy that are worth looking at. So what do the PET scans look like? On the top row is a normal. So on the left, you've got an amyloid PET scan, not too hot, and then it gets crazy hot for an abnormal scan. That would be an Alzheimer's patient. In the middle column at the top, a normal person on the tau PET scan, basically all blue is good. And then a patient down the bottom on abnormal tau PET scan in various hot parts of the brain, you'll have hotspots. And then in FDG, which is glucose hypometabolism, rather than measuring the actual pathology, you get quite high metabolism. The brain's using all the glucose well in a normal person at the top. And then at the bottom, you have an abnormal scan, particularly in the back left of the brain here on the image, where you get hypometabolism in the brain. So clinical trials have used these various different ways to assess thresholds of abnormality for inclusion. So that being said, These are pretty crude and pretty basic binary decisions, essentially. Are you in, are you not, based on some severities that haven't been working for a while, and people have started talking about sub-threshold accumulation of pathology. It's a term you may have heard. But it goes back to our lack of understanding of Alzheimer's disease. And just over a decade ago, essentially, maybe a decade and a half, there's some pretty intense debate in neurology about how Alzheimer's progresses. It starting with the syndrome, as we know it in living people. Starts from cognitively normal, progresses to dementia via this sort of prodromal phase of mild cognitive impairment. Pretty crude disease staging system. And then the hypothesis was that there's a sequence of dynamic biomarkers. They're not dynamic throughout the disease, so you can't just make one measurement and track disease progression monotonically. There's this sequence of changes. But as I mentioned, we found that there's heterogeneity in that. So imagine that these are actually different colors. Don't look at them too closely. They're just copies of each other. But the point is that to say that they're actually different versions. And this has been more recently formalized in the ATN framework, where you can have positive, tau positive, and various combinations of amyloid negative and tau negative, and neurodegeneration likewise. So you can have different subtypes where this sequence of changes occurs in a different order. And obviously not all those individuals would be suitable for the same clinical trial necessarily. So these are hypothetical models around about a decade ago that sort of summed up a decade worth of debate before that. But how does the data look? Well, in sporadic AD, this slide's credit to Mike Donoghue from USC. You've got on the left here the mini mental state exam, maximum score of 30, and declining as it becomes more blue. These are the clinical diagnoses on the bottom, orange to blue from essentially cognitively normal through to probable AD. You can see the scores declining. And this is over time, where the time axis is study time. So someone's recruited at time 0 and measured a number of years after. This is ADNI data. And the hippocampal volume, likewise, you have lower hippocampal volumes in the more blue phases of the later stages of clinical stages of disease as the hippocampus atrophies. But this is a mess. I mean, this is all relative to study time. This is not relative to the disease time. We don't know when onset was, all these things. So how do we understand this? How do I understand this? How does a computer scientist approach this? So we like to do something called data-driven disease progression modeling. Key element is that, unlike hypothetical models, it's informed by real data. And the second element is that we estimate, based on just a small group of assumptions, the actual timeline, or the pseudo time, which can be referred to. I'll get to that in a sec. Of the disease itself, as well as the trajectories. And here's an animation of that, courtesy of Marco Lorenzi's work from 2017, where you start with this scatter plot or spaghetti plot of just a few years of measurements across various biomarkers. And individuals can be shifted left and right, but all their biomarker measurements are shifted left and right, and you fit the trajectory to each one under that constraint. And you can get some visualizations like this. This is synthetic data. This is what the ADNI data looks like using the same model. So from Marco Lorenzi's paper again. And you can see that for some of the features, like in the bottom left, the FAQ, the Functional Activities Questionnaire, and the ADAS cog next to it, and even the hippocampal volume to an extent, match, fit quite tightly to an increasing sigmoid, kind of like a data-driven version of those Jack curves from Cliff Jack, the hypothetical model. But the others, yeah, not so much. Could be due to lots of things. Could be measurement noise, could be measurement variation, natural variation. It could be heterogeneity. Regardless, when you have someone moved along in the timeline, they're then assigned a probabilistic stage. So in yellow, we've got the normals. This is the cognitively normals in ADNI. Down through green into blue, where the probable AD patients are. And the model seems consistent with the clinical diagnosis. Most of the normals in yellow are staged towards the early part of the disease timeline, and most of the patients are staged towards the later part of the timeline. But now you've got this fine-grained understanding of the disease progression. You've got this more precise timeline to quantify the disease. But as I said, it's not perfect. There's quite a lot of heterogeneity in there. So what happens if you have, say, subtypes? Now let's go back to another cartoon version. So imagine you've got disease 1 or subtype 1 or disease 2 or subtype 2, disease 1 on the left here, and disease 2 on the right. For example, the key identifying factor here for subtype 1 would be that the green biomarker is quite late to become abnormal, whereas in subtype 2, it's somewhere in the middle. Point is, there's a different signature here for each of the diseases or each of the subtypes. You can take a single measurement from a new individual. The same biomarkers are in the same colors. Hopefully that's okay for colorblind folk. And then you line them up with the diseases, line them up with the signatures that you've estimated in a data-driven way, and calculate likelihoods on the vertical axis here going down. So for example, this particular individual has blue and red biomarker measurements that line up quite well early on in disease subtype 1. But at that same time, the orange and green markers don't line up. So it's pretty low likelihood that this data came from someone with this disease. On the contrary, in disease or subtype 2, the biomarker measurements for all the colors line up quite well at one of the later stages. And that's suggesting that this individual is a fairly late stage person having disease or disease subtype number 2. And this is a summary cartoon slide of the goal of data-driven disease progression modeling. Once you can do this precision staging and subtyping, then you can, and when we understand what happens after that, then you can find the optimal point and the optimal individuals at which to include in your clinical trial. So we can try to overcome some of this heterogeneity and find the right people at the right time for the trial. So the most recent innovations in this, and I do have another talk that I could give another time perhaps if you're interested, that gives the history of data-driven disease progression modeling in quite a lot of detail, starting from hypothetical models through to the 10 years that followed, through to sort of last year. But the latest thing is to add clustering. So we have discrete models that take the hypothetical model concept quite literally and just estimate a discrete sequence of dynamic biomarkers, and you can mathematically formulate that in a couple of ways or a few ways, and then add clustering on top of that disease progression modeling, which is the first time that clustering in at least in Alzheimer's disease has been added on top of a longitudinal signature, as opposed to just clustering a whole bunch of patients that you've got, which conflates the severity of the disease with the subtype. More recently, there's a continuous model that's come out, which is a latent time nonlinear mixed effects model, plus Gaussian mixture modeling. It's a nonlinear version of the workhorse of statistics, a mixed effects model. And it estimates a latent time, this is a disease progression model, and then adds a Gaussian mixture model to allow individuals to have some probabilistic assignment between subtypes. I won't dig into the details of that one. It was presented at an information processing in medical imaging conference a couple of years ago. I will focus more on subtype and stage inference, which came out of our lab. And then that actually evolved from the event-based model, which came out shortly after the hypothetical models did in 2010. The event-based model just aims to estimate that sequence of dynamic biomarkers in the pathophysiological cascade of Alzheimer's disease, and it works from just cross-sectional or short-term longitudinal data. There's no prior knowledge of disease stage. It shuffles these things around, and I'll explain how, without knowing what disease individuals are in. And it's flexible enough to allow for individuals to be in multiple stages. So this original paper came out actually in the same conference in IPME in 2011, but the journal article was in Neuroimage the year after. Hubert Fontaine and Danny Alexander put this together. And then a few years later, it was generalized and cleaned up a little bit to make it work for that. That was applied to familial diseases. You can see here familial Alzheimer's and familial Huntington's in Hubert's work. And then Alex Young, during a PhD while I was a postdoc, newly switched over from physics into Alzheimer's. And we did some methodological innovations that made it work for sporadic disease, which is a little more difficult. So quite a slightly busy slide, but let's have a look at the top left. We've got the familiar cartoon of sigmoids, two sigmoids, an early symptom and a late symptom. Let's call it a cough and a sneeze, just for concreteness. Now, early on, you have quite low measurements in both. So an individual individual measurement in a vertical column here on the bottom left has a yellow measurement for biomarker 1, the blue one, and a yellow measurement for biomarker 2, the sneeze, the orange. Someone in the middle has one yellow and one green. You can see quite a low measurement for the sneeze and a high measurement for the cough, the early symptom. And likewise, someone at the end has two green measurements, two abnormal measurements, high measurements here in the biomarker picture. Now, if you only have cross-sectional data, you don't have this time axis. So you can only have access to the histogram of these measures from 0 to 1. So on the left to right axis on this right panel goes from 0 being normal up to 1 with some noise on there. Now the green distribution is the healthy controls. So they'll have some noise, some variation around a normal measurement of 0. And the patients can be somewhere along the timeline. So they could have normal or abnormal measurements. So you get, say, the earliest measurement of the earliest event of cough, which we know is early from this cartoon. More of the patients will have high measurements closer to 1 in this blue histogram on the right. And fewer of them will have orange abnormal measurements. So you can see quite a few patients with normal measurements in the SNEEs, the orange histogram on the left. And that's also visualized by the shuffling around here of the individual slices now on the bottom left. We don't actually know where everyone is, and we don't need to know where everyone is because of this argument of if you've got one normal and one abnormal, then you know which one went abnormal first. And that can be applied across N features. For example, in the ADNI data again, this is from Alex Young's paper in Brain in 2014. We put in some CSF measures of amyloid and tau, some atrophy measures. So these were actually 2 time points converted into a rate, some cognitive measures in pink, and then some brain volumes in orange. And the density, the positional density here shows you the confidence that we have in this ordering. So a nice dark black one, dark black square indicates that we're quite certain that event was before the ones that come after, but there is some uncertainty in the others. And that could be due to multiple reasons. So loosely what we got was CSF protein measures become abnormal first, and then atrophy follows, followed by cognitive test decline and brain volumes declining. So when you line people up, imagine that cartoon that I showed before, but only one subtype and one disease, you get patients that seem to have lost their label in pink being staged towards the right. Cognitively normal individuals staged earlier, and the late and early MCIs are the orange and the yellow and green, and they're staged more heterogeneously across the disease timeline. So we've got this from cross-sectional data, we've got the sequence of abnormality in the pathophysiological cascade of Alzheimer's disease, and a tool to do more precision staging of these individuals. More recently, it's been taken up by other groups. So we had no knowledge of this paper until it came out. It's Oscar Hansen's group and collaborators over mostly in Lund, where they're interested in their new plasma blood biomarkers, particularly phospho-tau217. And they were looking at whether or not, part of their analysis was whether or not their plasma measures of tau lined up or became detectably abnormal before or after tau PET. So this is the familiar positional variance diagram on the left. And they showed that the CSF and plasma measures became abnormal before some interesting regions of interest in their tau PET scan, in the entorhinal cortex and in the meta region of the temporal lobe. And then they produced some statistical trajectories as a function of the model stage. So the event-based model is EBM. So that made us happy because we're trying to help. And we put the code out a long time ago, and it's starting to be used now, which is great. So the event-based model inspired SUSTAIN, the subtype and stage inference model. So this is the implementation of our cartoon from before, where we try to subtype and stage individuals at the same time. So this is a figure of how it would work. If we make an assumption that there's an underlying model of some increasing atrophy, so it looks like it's spreading across the brain in different patterns, different ways, spatial patterns, spatiotemporal patterns of atrophy. Let's say, for example, for argument's sake, there's 2 subtypes of atrophy. What you actually get is this heterogeneous snapshot. You don't know which individual came from which subtype. But using the same logic as before with the event-based model, you can combine individuals and look at you can think of it as looking at the frequencies of abnormality across regions. And you can learn what these subtypes are. This is the bottom right panel. So SUSTAIN reconstructs these disease stages within subtypes. And then you not only get a stage, as I showed you just before for the event-based model, which is along this top left to bottom right x-axis in the histograms on the top right panel, you also get a subtype fully probabilistic, so individuals get probability for each stage and for each subtype, and you can make a hard cutoff to make any decisions based on that to give them a subtype and a stage. This is published, this is Alex Young's work from Nature Communications in 2018. The process is to sort of divide and conquer. You tell it how many subtypes to fit, and that's quite common in clustering algorithms. And then we use cross-validation to select the final number of subtypes that are consistent across multiple runs of cross-validation. So one of the coolest findings of the original sustained paper was in the GenFi dataset, genetic frontotemporal dementia, where all these individuals have one of three families of mutations: progranulin, GRN, MAPT, and C9orf72. So the model found 4 subtypes. The data could support 4 subtypes, which were described loosely by the earliest events in the atrophy pattern to be asymmetric frontal, temporal, frontotemporal, and subcortical. And when you do the subtyping and staging, and then just plot histograms of the genetic mutation family that the individual's work came from when they were subtyped, which is not information that went into the model, all that went in was MRI volumes, then you find that the asymmetric frontal subtype was primarily progranulin mutation carriers. The temporal subtype was primarily MAPT, and the frontotemporal and subcortical took about half of the C9orf72s each. And apparently that was known to be a heterogeneous mutation with a different, with 2 predominant phenotypes that come out in the clinic. That was a really nice finding. Alex also applied it to ADNI, both to the 1.5 Tesla and to the 3 Tesla scans, and found basically 3 subtypes: typical cortical led, where the hippocampus doesn't atrophy until a little later, and the subcortical, where the cortex is left until a little later. And they had slightly different clinical phenotypes. The cool thing about it is if you do some subtyping and assign someone the probability of being in each subtype as this position on a triangle, if you focus on the cognitively normal triangle to the bottom left to start with, then if someone's brain MRI aligns very, very well with the typical subtype, even if they're cognitively normal, then they will be right around this apex of the triangle. Likewise for the cortical down the bottom left and the subcortical, the bottom right corner of the triangle. And those in the middle have some probability of aligning with all 3, but don't strongly associate with any one of them. But even from the cognitively normal stage, you can see that some individuals were strongly identified in the subtypes. As you get further down the clinical line, probable Alzheimer's disease patients, their atrophy patterns more and more match the subtype quite well. So this means you can do things quite early and potentially pre-symptomatic with this particular model. So the highlights for further reading then, you may have heard of Jake Vogel's recent paper where he assembled a whole bunch of tau PET data and found 4 subtypes of fairly common subtypes of Alzheimer's disease, of tau deposition, in Alzheimer's disease, where they were roughly 20-25% of the population each. Arman Ashagi from UCL assembled something like 13,000 MRI scans from clinical trials in multiple sclerosis and identified 3 subtypes of multiple sclerosis, 1 in particular, but 2 of which responded to treatment in these clinical trials, and that wasn't known beforehand. So the model seemed to be able to identify potential responders without information beyond a couple of MRI. And the original paper is Alex's. If you're interested in running the code itself, it's available. It's been available for a while, and we've actually got a Software X paper that describes the code and exactly how it runs. That's led by Leon Axman, who's now in Art Toga's lab, I believe. And then there's actually another group that I haven't got the paper up here. Lidwine Colley et al. From the Amipad family has found 3 subtypes of amyloid deposition across multiple cohorts. Again, like Jake, she assembled a large cohort of individuals at risk of Alzheimer's. So my desire to make a difference has focused on me not developing many new models in the last couple of years, but trying to apply these models in clinical trials and try to put my money where my mouth is, although someone else's money is strictly. The challenge is to get hold of data from clinical trials. So I've spent a lot of time talking to people and convincing them to collaborate. And we've had some success. So I do have some results to show you, which is quite heartening. We've got some event-based model results, and they fairly recently came out in Frontiers, although the paper was submitted in 21. That's using the event-based model in the MCI trial, which some of you may be familiar with, donepezil and vitamin E from a number of years ago. And more recently, with my postdoc Cameron Shand, we've been working on some more recent clinical trial data using Sustain, and that was from the FIN trial. I'll show you the results of both of these studies. So the original NEJM paper in 2005 for what we call the MCI trial, which came from the Alzheimer's Disease Cooperative Study, probably very, very close to where you guys are. It was a trial of donepezil and vitamin E in MCI. So the experimental recipe is to build a model. So I build a model on ADNI data, say, and then I use that model to stage and eventually, if you have sustained subtype, the trial data at baseline, just with that cross-sectional data. Stratify somehow, and then look at this as a subgroup analysis. This is often done in clinical trials. They have planned post hoc subgroup analyses, but they don't use disease progression models to define those subgroups. They'll use things like, let's look at just ApoE4 carriers, an increased risk of Alzheimer's disease. Let's see if the drug worked for them. Let's see if it worked in men or women, if that's a hypothesis they have. So the same sort of idea, but this time we use a disease progression model to do the stratification. The MCI trial didn't have much MRI in it. So this is not an image-based model. This is all cognitive instruments, ranging from clinical dementia rating through memory tests, MMSE, some ADAS-Cog, other recall tests, and some executive function. It came up with quite a clean sequence of decline. I'll note that although I haven't got the data here, the difference between these events were kind of subtle, although not quite as subtle as the treatment effect in monoclonal antibody trials. So this is the trained model. You train the model and you stage the ADNI data here. So the ADNI data went into train the model, found that sequence, and this is the staging of the ADNI data. So we've got all the cognitively normals in blue, stage 0. The MCI in green are staged fairly flat distribution across all stages. So some had abnormality only in clinical dementia rating and only subtly. And some had cumulative abnormality in all the instruments up to the sort of second last, I think, which was Boston naming. And the Alzheimer's disease patients, probable Alzheimer's disease patients in ADNI in orange, were all staged past the CDR events with a skew towards later stages, as you might expect. So then I took the MCI trial data and staged those as well and found this interesting trimodal peaked distribution of early, middle, and late cognitive decline, you would call it, according to the model. And I thought, oh, well, it looks peaked, so let's do a subgroup analysis on the three subgroups. So you got about 360 in the first less impaired subgroup, about 220 in the middle, and 85 more severe. So if we split those down then and look at this severity score of less than stage 5 in the top row, the 362, and then we look at how their treatment went in donepezil and vitamin E. These are two separate arms in the trial. And then we score at each of the 6 monthly intervals in the trial, 6 to 36 months, the difference in placebo versus treatment in the primary outcome, ADAS-Cog 13. So a negative difference here would say that the treatment has a higher score than the placebo, and that's worse in ADAS-Cog. So negative means that the treatment arm didn't do very well. Ideally, you want a positive score here. So I've highlighted the 2 points at which the treatment showed some effect. Obviously, it wasn't maintained for the full period, but it was shown in the donepezil arm of individuals at the more advanced stages of cognitive decline. So perhaps these were the ones that were primed and ready to show some decline. So the placebo arm, maybe shot off to higher values of ADAS-Cog 13. But you can see this practice effects, learning effects, noise of some kind in there, where it fluctuated. And even when you have a difference of 4 on the ADAS-Cog here at 24 months in the donepezil arm of the severe group, it wasn't statistically significantly different. That was quite promising. And then more recently, we got hold of the FIN trial data. So the idea here was that this would help reduce hypometabolism in the brain, which would then lead to improved outcomes from less neurodegeneration, et cetera, et cetera. So again, build the model on ADNI data. This time we found two subtypes, a subcortical and a cortical. It's a very basic model, these are two event-based models essentially. So the first model follows this diagonal, follows the sequence on the vertical axis, which is fairly typical Alzheimer's. You got some limbic regions, hippo, amygdala, entorhinal cortex, temporal lobe. But then subcortical regions seem to get involved before the rest of the cortex. Whereas some of the earlier events in subtype 2 were the cortical regions. So we named these subcortical and cortical. Approximately, not quite 2/3 to 1/3 ratio in the population in the ADNI data. So these were MRI volumes again. I think they were calculated with FreeSurfer. So in the ADNI cohort again, we subtyped and staged. The subtype is green, and the subcortical subtype is green. The cortical subtype is in orange. These are next to each other within each stage of disease. So then the density was quite high early on, and then fewer individuals in later stages. And when you do that in the FIN cohort, the trial cohort, which was about 150, I think, individuals, a few didn't survive because the image processing failed. So we ended up with about 50-50 split in the subcortical and cortical subtype. And it seemed that the cortical subtype had quite a few individuals with more cumulative abnormality. So they were staged a little bit later. There's quite a bit of heterogeneity in this clinical trial. So we kind of weren't surprised when we looked at outcomes. On the left here, you've got ADAS-Cog 12. So increases is worse. Placebo is orange. So if you look at the cortical subtype, you can see that the placebo group had higher scores, higher change. So this is actually change over the 52 weeks of the trial. They had a higher increase than the treatment arm in blue. And on the right-hand side, the primary outcome of this phase 2 trial was the FDG PET change in a statistical ROI, which is a voxel-wise statistical method to maximize signal between MCI and controls cross-sectionally. It might have been AD. There's two versions of it with AD versus controls and MCI versus controls. And you can see that there's no real discernible differences in either of the subtypes, nor in the group as a whole on the left. So it looks like maybe there's some cognitive benefit, but not much difference on the primary outcome. And this, I believe, this development of this drug has halted. So we looked at some other outcomes as well. So these are all MRI now. Hippocampus at the top, percent change, so lower is worse. Lateral ventricle change, so higher is worse. Whole brain change, so again lower. Entorhinal, again lower. The first on the left, you see not much difference in within the whole trial cohort between placebo in orange and treatment in blue. Some potential differences you're looking maybe. But if you look at the cortical subtype, or the subcortical subtype actually in this case, had more atrophy in the hippocampus. The cortical subtype seemed to have slightly higher atrophy in the ventricles, and in the whole brain, and in the entorhinal cortex, although the subcortical also had a trend towards that as well. So the take-home messages then, we've done a couple of different models in a couple of different clinical trials, certainly found some heterogeneity, which correlates with our increasing acceptance of the knowledge that Alzheimer's is a heterogeneous mystery. And I would argue that what I've shown you is that disease progression modeling can potentially help to unravel this mystery. And disease progression modeling particularly produces these quantitative templates that you can then line up individuals with to use for something like enriching clinical trials to find the right patients at the right time. Lots of caveats on that, obviously. So you do subtyping. Let's imagine that there are 4 subtypes and the fourth subtype represents 10% of all Alzheimer's patients. Hard to convince a clinical trial sponsor to run a trial just on 10% of the patients, especially now that it seems that their broad spectrum approach might be starting to work, reference lecanemab. But still, so many successes and so many failures over so long. At the very least, what we could do is post hoc go back and look at these failed trial programs and see if there might be some hope for some patients for some of these drugs. Once you've done that, obviously, these drugs get onto the market and they get into healthcare. So you need to help with healthcare and help to inform who can be prescribed the drug. For example, if you use a model to select for a trial, you probably need to use the model to inform prescription. With that in mind, I've been reaching out to healthcare as well, particularly to a local memory clinic in England, just to see what their data was like, just to have a little play, see if we could train some models. And one interesting thing that we found out, so this is the only slide I have, and this is not much here, is that in this particular memory clinic, so lots of different dementias coming in, the clinicians are trained to use the T2weighted MRI. So they like to use the T2weighted MRI for routine care. It's fairly thick slices. You would call it a 2D scan rather than a 3D scan. It does cover the whole brain. And they use it for looking for lesions and for also the visual rating scales in different parts of the brain. So because it's quite a low resolution scan, every different modality that they do take a few modalities, gets the same amount of time. And the killer is that the default resolution for the particular scanner they use for a T1 is higher, so it takes longer. And the result is that you end up with a cropped image. You don't have the lateral temporal lobes involved in the scan. So we have some preliminary results that will be submitted soon. We're presented at AAIC this year in a poster, where we use the T2 scan in conjunction with the SynthSR tool in FreeSurfer to fill in the cropping side. And we show that we can reduce bias and recover fairly good estimates by starting with ADNI data, artificially cropping it. So we did have a ground truth, and then running it through our pipeline and estimating the volumes. And then we saw the downstream effect of that on the disease progression models as well. Always end with a big thanks. All the research participants, without their data, computer scientists wouldn't even be interested. Their families, who obviously do a lot of the work to get individuals to the data collection sites, and all my colleagues, in particular in the POND group, but also all the clinicians that I work with at the Dementia Research Centre in our joint quest for supermodels and drugs.